with Punnett squares. And here's where we left off, right? We had this last time. Oh, let me let this person in. So we assign genotypes to our classic Mendelian experiment here. We assign genotypes to our various generations, and we discovered that the recessive white pigmented flower disappeared in the F1 generation. It was sort of hidden, right? In these two heterozygous carriers, heterozygous genotypes. And we call them carriers because they carried the recessive white allele. So when we cross them, you actually saw the re-emergence of this trait. And so this sort of crossing and combining genotypes in two parents that are going to be mated and have offspring, we do this technique, as I left off, called the Punnett square. Punnett square, right? So we're gonna practice that right now. Okay. So two parents who are normal height have a child with dwarfism. How is this possible? So it's kind of a similar situation here. It's kind of like two purple flowers had an offspring with white pigment. The parents didn't even exhibit this, or how we say, express this trait, this phenotype of white flowers. So how did it happen? I'm gonna kind of just walk through it with you guys. Get my chair out of the way. Okay. I think it's achondroplasia, the condition. Okay. So the first thing is we have to determine the genotypes for the parents. And if they're both normal height, and let's just say big D is going to be our dominant allele, and it's a normal height allele. So essentially this condition is a recessive condition because little d, the recessive allele for dwarfism is going to be little d. So dwarfism allele. And I'll add normal height allele. Okay. Now, the problem told us that both of the parents are normal height, right? So that means no parent is going to have the genotype little d, right? That's the only, that genotype would give the dwarfism phenotype. And essentially the only way that two parents that without this recessive condition can have a child with the dwarfism phenotype is if we did the Punnett square like this. And this is how you do a Punnett square. You draw this square with 4 squares inside the square here. And you put the genotypes of the two parents on the top and the left side. And I'm going to go ahead and tell you that the two parents have the following genotypes. Where did I put my markers? Here we go. The two parents have the following genotypes. Big D, little d. Actually, I'm going to move it. I'll move it to the side here. We're going to cross big D, little d with big D, little d. They're both heterozygous for dwarfism. Heterozygous. Okay. Now, if we know these are the two parents and we're crossing them, we're going to put one of them here like this, and then the other on the left side here. Then we go over and down, depending on what square we're at. So for this square, it'll be this D comes down, this D comes across. For this square, this D comes down and this D comes across. So it'd be big D, little d. This guy's going to come down, big D. He comes across here. And then these two. And so here is our recessive genotype for dwarfism. And essentially what the Punnett square tells us is that there's a 25% chance that their offspring here, it's a probability. Punnett squares give us a probability of genotype in the offspring. So it's a 25% chance child has dwarfism. And of course, all of these genotypes, since it's a dominant relationship, normal height's dominant. And remember, we denote dominant recessive as uppercase capital letters versus lowercase. But that would be a 75% chance, 3 out of 4 chance of having normal height. Okay. Just another side note. Oh, they have to be both heterozygous. Correct. So I'm going to actually talk about both parents. Correct. Sana, that's correct. That both parents don't have dwarfism because they're heterozygous and the dominant allele is normal height. So remember, it kind of overshadows the recessive allele. So you won't see a recessive phenotype unless you have 2 copies of the recessive allele. So before I address Christine's question here, we sometimes call heterozygous individuals carriers for that condition. So the parents here are carriers of dwarfism. They carry the allele, right? So you could call these carriers. Just in case you run into that word, they're carrying the allele, but they're not expressing it, right? They're still normal height. Now, the reason that their genotypes have to be like this, as Christine mentioned, do they have to be heterozygous, both of them? I'll show you. Let's say one of the parents was homozygous dominant for normal height here. Big D, big D. I'll show you what happens. So one of the parents now has a genotype, big D, and I'm going to keep the other genotype heterozygous for the other parent. But when I do my Punnett square, you'll notice there's no 0% chance of dwarfism now, right? There's no little d genotype possible. So no matter how many children those parents have, there's no possible chance they'd have a child with dwarfism. And really, if you look at it, if you have this homozygous dominant parent, both columns are going to get at least 1 big D. So there's just no way that a child could have 2 little d's. So to answer your question, yes, they both have to be carriers if both are normal height in order for them to have a child with the recessive trait. Okay, so now there's another really important point here that touches upon this probability concept for number 2. True or false? If the normal height couple had 4 children, 3 would have to be normal height and 1 would have dwarfism. What do you think? True or false? Nice. Yeah, false. It seems like you guys got the concept. So I'm going to say for that one, it's only a likelihood. It's a probability, right? So we said that it was, right, it was 75% chance normal height, 25% chance dwarfism, right? So 4 children, it does not have to be 3 of them normal, 1 dwarfism. Every time they have a child, it's that same Punnett square probability. So every time they had a child, it would be 75% chance normal, 25% chance dwarfism. So yes, it's possible that all 4 have dwarfism because every time it's a 25% chance. And we won't get too math crazy here, but that's pretty rare. It's 1/4 times 1/4, whatever. But I want to just emphasize the point that it kind of resets every time. It's a likelihood. It's not set in stone. It's not a hard, fast, yeah, 3 out of 4 will be this and 1 out of 4 will be that. It's kind of, the analogy I like to use here is if I flipped a coin, heads or tails, 4 times, it doesn't mean 2 out of 4 will be heads and 2 out of 4 will be tails. No, I could get heads every time. I could get heads 3 times. I could get tails 3 times. Same kind of thing. It's a reset. It's a probability each time they have offspring or a child. Okay, so let's do a fun in-class assignment here, a little more practice with this. In groups of 2 or 3. Just ignore that because we're not in person. Okay, so freckles is determined by the dominant allele, capital F. So I'm actually, it's a little vague, but what I mean is freckles is a dominant trait, meaning if you have the dominant allele, at least one, you will have freckles. So go ahead and in your notes, take a minute or 2 to write the genotype or genotypes for each of the following phenotypes. And then answer, is it possible for a freckled child to be produced by 2 parents who are not freckled, and then explain why or why not? So go ahead and work on that for a few minutes. I'll leave this up actually. Give you one more minute. Oh, my voice is gonna crack. Great. Hasn't happened in a while. Give you one more minute. Okay. The genotype or genotypes for following phenotypes. Freckled. What are our genotype or genotypes? Christine is correct. Remember, genotype, yes. Genotype is 2 alleles, right? So freckled, you're gonna have, I'll go to the, freckled would be big F, or big F, little f. Exactly. Non-freckled? Only one possibility. It's recessive to be non-freckled. So little f, little f. So this is freckled. This is non-freckled. Okay, is it possible for a freckled child to be produced by two, why did I use that terminology? Whatever. To be born, whatever, I don't know, by two parents who are not freckled? Yes or no? And then I'll kind of talk about it. Ooh, we gotta be careful. No. Yeah, it's no. So Joey, if two parents are not freckled, then they would both have FF. That's correct. Exactly. So we're going to set this up. Not having freckles is recessive, right? It's non-freckled. And both parents are non-freckled. So let's do it. Right? And then all you got are little f's. This kind of explains why certain recessive traits can kind of stay within families who marry other similar recessive trait people or have recessive traits as well. Like two parents with blue eyes, for instance, you don't really get a brown-eyed kid, at least not very often. So it has to do with this sort of concept. Okay. Well, that was it for genetics part 1. Making really good time. Like I said, after we go over part 2 today, some of the questions in the lab will make a lot more sense. Wait, but the blue-eyed parents can be carrying the brown. That's correct, Sana. Yes. Yes, it's true. However, that's only if brown is recessive, right? So let's say eye color is, we'll use B as the allele. And if big B is dominant and brown, little b is the recessive allele for blue, the two parents with blue eyes, the only possibility of their genotype is little b, little b. The only way they could be carriers, well, actually, they can't be carriers and have blue eyes if it's recessive. The only carrier situation for these parents would be parents that both have brown eyes and are heterozygous. And then in that case, you can get a kid with blue eyes. And then you're like, whose kid is this? Kind of thing. Okay. What if both the blue and brown parents are homozygous? Let's find out. Blue-eyed, yes, no, they're not blue people running around. Let's try, let's do it. So big B, little b, little b. So they'd all be heterozygous for eye color, or brown eyes. So they'd all have brown eyes no matter what. In fact, really quickly, this is the same situation as the initial flower color setup I gave you with the purple flower that was homozygous dominant. Oh, did I use P, right? Big P, with little p, because it was crossed with a white flower. And then that F1 generation was all purple flowers, right? No matter how many offspring Mendel produced with these two flowers, he'd plant 600, 6,000, and they'd all be purple. That white recessive trait never showed up, and this is why. 4 out of 4 here, 100% possibility every time of being purple phenotype, because you have that dominant allele. Good question. Okay. The next thing I'm going to talk about, I like to go back, right? I like to draw back on previous concepts because it's kind of like, why did we learn it if it's not super relevant to other things? And I like, I always wondered in school, like, how will I ever use this, right? It's like, why am I learning this? I'll never use this in my life. In biology, everything's related. So no matter what you learn in biology, it'll apply to another concept and be important. It's part of why I love it. So genetics part 2. We're going to do a 2 trait cross. Actually, before that, we're going to do a fun drawing. Drawing back, a drawing that draws back on a previous concept we talked about, meiosis. And I'm going to use, I'll use green, sure. Okay. So we're going to take a look at 2. So far, I'm about to make things more complicated, sorry. But so far it'll make sense though, I promise. So far we've been looking at one gene at a time. Height, we looked at flower color, we looked at, what's the other one we just did? Blanking. Height, flower color, is that it? Freckles, thank you. Freckles or non-freckles, that's one gene for each one, right? What if we want to do 2 at once? Now remember, every gene had 2 versions. So freckles, for instance, it was freckles or no freckles, 2 versions, dominant, recessive. Flower color, purple or white, 2 versions. Height, normal height, dwarfism, 2 alleles per gene, right? What if we did 2 genes at once? Maybe there'd be 4 possibilities. So let's take a look here. we're going to do 2 genes. And remember, we're actually going to be doing not just 2 genes on 2 different chromosomes. So let me draw what that looks like. In a cell that's going to undergo meiosis, okay? I'm going to do 2 homologous pairs. Of course, we know there's 23 pairs. And so this cell here is going to undergo meiosis. And remember, in metaphase, the homologous pairs line up like this, right? Side by side. And we're going to do 2 genes at once with 4 different alleles. So I'll show you what that looks like. First, let me just say the genotype for this individual will be big R, little r, big Y, little y. I'll move this up a little bit. And in a moment, we'll talk about what these alleles will be for. But the best example is going to be with pea plants like Mendel did. But if the genotype, so I like to do this because it connects why things are the way they are. And when why there's 2 alleles for a gene. So the genotype, Amber, it's actually 4 alleles in a row for 2 genes. Big R, big Y, little y. And it looks like this. The big R is located on this chromosome. Remember, same gene can be a different allele, right? Same location on, and this is for homologous chromosomes. Should have prefaced with that. The homologous pairs, like the 2 chromosome 1s or 2 chromosome 2s, have the same gene, but it can be a different allele. And that's exactly what's going on here. Big R, little r. Same gene, but there's a different version of that gene. There's a big R on this chromosome and a little r for the same gene on this chromosome. And then down here, big Y, little y. 2 genes at once on 2 homologous pairs here. 2 different chromosomes. Okay. So let's see what happens as we go through meiosis. But I just want to say one more time that when I saw in a textbook alleles placed next to their homologous chromosomes like this, a light went off and I never forgot it. It explained why the meiosis process and why Punnett squares, why there's 2 versions of a gene, and why when we do meiosis, a gamete's going to contribute 1 allele from these chromosomes. You're going to see that in just a second. So let's do 2 cells. This is after meiosis 1, right? So if we divide that down the middle, this cell is going to have big R, big Y, right? And then this cell is going to have little r, little y. And then, of course, so this was meiosis 1. And then meiosis 2. This is when our sister chromatids separate to create identical cells. So that's just going to be, so this is big R, little r, big Y, big Y, little y, sorry, and little y. Okay. So far here, we've got two gamete cells. Maybe we're making sperm cells, male, or egg cells, female. Two here that are big R and big Y. And then we have two here that are little r, little y. They're the same, right? These two and these two. So I'm actually going to write below here possible combinations. And we have a gamete with big R, big Y, and a gamete with little r, little y. But that's not all we have, is it? What was it that we can do in metaphase 1 to increase genetic variation, to get all those combinations? Not crossing over. Yes, crossing over plays a role in prophase 1, but it's the way that our chromosomes line up in metaphase. What was that called? We got the trillions of combinations just by how our chromosomes lined up. Yes, it's called independent assortment. So this is what I'm going to do. I'm going to switch these places here. I'm going to switch this chromosome over here and that chromosome there. I'm going to swap spots. Sorry, what are the black? Oh, those are chromosomes. So these lines, those are single-stranded chromosomes. Okay. Oh, so I switched those spots, right? The big Y and little y. So I'm actually going to switch them for you. So I just switched them. And now, when I divide this cell in half, I no longer get big R and big Y up here and little r, little y down here. I get little r, big Y, little r, big Y, and then here would be big R, little y. And then, of course, that's going to produce this big R, little y for both of these, and then little r, big Y for these two. Essentially, the point I'm making is we have now two more possibilities for gametes, depending on where I swapped one of them. So if I swapped them, I now can have a gamete with big R, little y, as seen here, right? And then little r, big Y. So you get 1, 2, 3, 4 possibilities. If you're working with two genes on two different homologous pairs. And so we're going to do what we call a dihybrid cross, where we're going to cross same Punnett square type cross, but it's bigger because we have two genes at once instead of one. Is this the thing where you do 2 to the n? Yeah, kind of. We sort of did that already when we did 2 to the 23rd. But you have to remember something crazy, but there's anywhere from 100 to 1,000 genes on a single chromosome. But yeah, so there's a lot of options. Okay. But yeah, I'd like to draw it out like this just to connect it to meiosis. Because it kind of, I don't know, when I learned it, it was like, why are we doing this? The two alleles, the one gene that's, you can have two variations. This is it. You have two variations possible on your homologous chromosomes. And it gives you, you're going to see why the probability, because of independent assortment, why we're doing these Punnett squares, why they can be predictors for us in genetic outcome of an offspring independent. Should 4 be big Y, little r? It is. It is. I did little r. Oh, I get what you're saying. That doesn't matter. And I'll tell you why. Because they're two different genes. They're two different genes. And so when we do our dihybrid cross, it'll make more sense, I think. But for a specific gene, I get what you're saying. You'll never do like little r, big R. That's one gene because it's the same letter, but it would be big R, little r, right? If you have an R and a Y, two different letters, that doesn't matter. Okay. So this is what we're gonna do. We're gonna do a dihybrid cross. We're crossing two trait cross. So if we look here, we're gonna be looking at two genes at once. P shape, so plump versus round. And then sorry, round versus wrinkled. Those are pods, just ignore that. We're gonna do these two here. We're gonna do the P's themselves. This was just Mendel, a picture I took. P shape, round versus wrinkled, okay? And then we're also gonna cross it with P color, two genes at once. Okay, so we're gonna do color, green or yellow, and shape, round or wrinkled. Okay, let's do it. We're gonna cross two parents with this genotype. Or these genotypes. They have the same genotype. One is like this, the other is like that. Heterozygous for both genes. Both of them are. Heterozygous for P shape and P colored. So I'll just quickly mention that round will be dominant, wrinkled will be recessive. And so when we get the outcome, I'll write this down. And then, yeah. Oh, for color, what was it, yellow? Yellow is dominant and green was recessive. So when we do this cross at the end with the outcome, I'll show you, I'll write that down. Okay, it's gonna be a big fun cross here. Now we just learned that one parent with that genotype is gonna be able to produce 4 combinations. And let's just say sperm, okay? 2, 3, make this a little bigger. So one parent would do big R, big Y, big R, little y, little r, big Y, and little r, little y. Those were possibilities. So let's make these sperm cells. just to remind us that sperm and egg come together to form an offspring. And then if the female has the same genotype, same gamete possibilities, right? Big R, big Y. Big R, little y. Little r, big Y. Little r, little y. And so we're going to have a bigger cross to do. This black is dying on me. Okay. So this is the part of my lecture where I'm kind of quiet as I fill this in. So just fill it in with me. But they're going to come across, right? And down, down and over. I think this is kind of fun. It's meditative. That's correct, yeah. Oh, wait a second. Yeah, that's right. Do, do, do. Okay. Okay. Woo! Isn't that great? So what are our possible outcomes here? Well, I'll just give you the spoiler. Whenever you cross two parents that are heterozygous for both genes in a cross like this, so heterozygous for shape and color, and they're both the same, right, genotype, you get the ratio 9, 3, 3, 1. So 9 of these genotypes, 9 of them will give you the phenotype round and yellow. Remember, round was dominant. So you're going to find 9 of these with at least a big R in it, at least one. You're also going to find 9 with at least, because yellow was also dominant, at least one big Y. 3 of them are going to be round and green. So the recessive, you're going to find 3 that have the dominant shape and then the recessive color. There's going to be 3 more that are wrinkled and yellow. And last but not least, you're going to have 1 out of the 16 that has both recessive traits, both recessive phenotype. It's actually this one here, little r, little y, wrinkled and green. You get that 1 in 16 chance that the offspring will be a wrinkled green pea producer plant. So we call this a dihybrid cross. And you're doing the 2 genes at once. Now, the lab will address this. It'll also address another thing where not all the crosses with 2 genes are going to be this hectic with 16 squares, 16 possibilities. So we're going to do another, yes, but it's very, very easy. believe me. Don't worry. We're not going to do another giant cross. Can you imagine if I did 3? What is that going to be, like 64 squares? Like craziness. Okay. The next cross, let me see how much we have. Okay. Oh, 30 minutes. Okay, I think we'll finish right on time. Okay, the next cross is going to be another dihybrid, but we're going to do a different genotype for our parents. Is anyone still writing? Okay. Sana, you're good? Okay, cool. No one? Really? Sweet. Kept up with me. All right. So let's do one more. It's super not as crazy, trust me. If you are still writing and you didn't want to type, or you went to the bathroom or something, it's recorded, so that's nice. All right. I think so. All right, so we're going to do another one, but this time this is going to be the genotypes of our parents. It's going to be, no, this was the worst one. Big R, little big Y crossed with little r, little y, little y. So the key here, after we saw the whole meiosis process and independent assortment and what kind of combinations we can get in gametes, this genotype can only give us 2 possible combinations, 2 gamete combinations. So what are they? If you have big R, big Y, it's only 2 options here. Well, you know you gotta give a big Y, right? Because there's only big Ys. Yeah, exactly, Sana. Big R, big Y, or little r, big Y. There's no other possibility, right? How many can I get from this one? How many combinations? Little r, little y, little y. Just one, exactly. It's only one possibility because if you think about it, independent assortment, no matter how you switch them, in meiosis, you're still getting big R, big Y, or little y at the end of the day because they're the same. And so if we do this cross here, let's do big R, big Y, little r, big Y. Say this was our sperm cell and our egg cells. And then you get that's it. We're done. Better, right? So it's a 50-50 chance of having, this would be round and yellow. And then this is wrinkled, right? Recessive and yellow. No matter what, you get yellow, but 50% chance of round or wrinkled. So they're not all super scary and awful giant crosses. There is a question in the lab that asks about gamete possibilities with genotype. So that's how you do it. And I believe there's also a 16 dihybrid cross as well. Okay, so we're going to step back from doing crosses and practice problems to doing some just general terms, definitions. What time is it? Oh, we're doing great. Any questions, actually? Just pause for questions. Okay. Okay, let's talk about what a polygenic trait is. Just the definition. I haven't been using many colors today, so let's do blue. Let's do blue. Okay, yeah, take a look at that slide. Okay, so polygenic trait. If something is polygenic, it means more than one genic gene. So multiple genes, okay? A polygenic trait is a trait, a single trait, such as skin color, height, eye color, actually, these are polygenic traits that are determined by more than one gene. So I'm going to write sometimes, or actually rather, let's see. A polygenic trait is one phenotype determined by a combination of more than one gene. And a few examples, like I mentioned, height is determined by a lot of genes, kind of like genes involved in bone growth and muscle growth and skin growth. Lots of genes involved. So human height. Skin color, pigmentation is determined by more than one gene. Eye color. Yeah, so these are just a few examples of polygenic traits, single traits determined by more than one gene. Okay. So the next thing is the opposite of this. It's called pleiotropy. Pleiotropy is the opposite. It's when one gene determines multiple traits, multiple phenotypes. Okay, so we're going to write that down. So I'm going to write So pleiotropy is when one gene determines multiple phenotypes. More than one, multiple phenotypes. And a classic example is albinism, as shown in the slide. And mutation in one gene, a pigmentation gene, results in the loss of pigment, as you can see in the skin, the hair, and the eyes. So multiple traits are affected by a single gene. That's pleiotropy, or it's a pleiotropic effect. That's sort of the classic example. Another example, that's right, I was going to think of. There's actually a gene on the Y chromosome for males, XY. The Y chromosome, it's called the SRY gene, is hugely important gene in male development. It regulates other genes and it causes the development of facial hair and other male associated phenotypes. But at this one SRY gene, it's just like this really crucial gene that controls a lot of that male development and those traits. And of course, females don't have a Y chromosome, so they wouldn't have that gene. But this is pleiotropy. It's multiple things being affected by one gene. Oh, sorry, let me erase this. That was for polygenic. The examples would be albinism. You could even say male development. Okay. Why some male, I got a question. So why some males don't grow as much facial hair? You know, I honestly have no idea about that. Oftentimes, especially in genetics, things are a lot more complicated than you think. And so, yes, there's the SRY gene, but I think that, that has to do with testosterone production, has to do with probably other genes. There's always, because, we have, 20,000 genes. It's always a more complicated story than just like one SRY gene. But I think a defective SRY gene can result in like serious developmental issues. Or one of the definitions talk about the reason for why twins don't sometimes look like. So sometimes twins don't. So that has to do with fraternal twins versus identical. Identical twins do have the same DNA and genotype. That's why they're identical, right? But fraternal twins are actually, they have completely different genomes. They have completely different DNA. Oh, it's a lab question. Yeah, I think that was the answer. So yeah, the fraternal twins, they just happen to be born at the same time. Kind of like my brother and I were born at the same time, but we're completely different people. That's like a fraternal twin. Yeah, exactly. They have different genotypes, different sets of genes. Yeah, totally. Okay. So this next example has to do with codominance. It's another term that we're going to learn. So it's blood type. It's kind of the classic example of codominance. And codominance is when two alleles are equally expressed slash, I'll write share dominance. So in a situation that's codominant, so really today's lecture, as you can probably tell by now, really focusing on variations of classical Mendelian genetics, the classic dominant recessive relationship, where if you have one dominant allele, you're going to see that phenotype. And you can only see the recessive allele if you have both recessive alleles, right? 2 copies. You're only going to see that trait if you have 2. But today's lecture is sort of little exceptions, little twists and turns in that classic story of sorts. And one is codominance, when 2 alleles are both expressed equally. There's no masking of the other allele, like dominant recessive. They're codominant. And so blood type's a good example. And so I'm going to write the 4 blood types. Type O, quotes me, I'm just writing the same word, type A, B, and then blood type A, B. Just want to make sure I have time to, okay, I will. Let me see. Okay, what is that, 15 minutes? Okay, I think we'll be okay. I'm hopeful. So these are the phenotypes, right? You have type O blood, type A, B, or AB. So the genotypes, what this is actually talking about when you have a different blood type is the presence of these cell surface markers. So you can see here for phenotype, if you're type A, you have these A sugars on top of your red blood cells, coating them on the surface. B, you're going to have B sugars, but then you'll notice AB people, some of you may be type AB, have the sugars A and B present. This is a codominant situation. A and B are both equally expressed. There's no overshadowing. There's no one or the other, right? And then, of course, type O is none of the above. No cell surface markers. Actually, type O is considered the universal donor for another story. You'll learn more probably in micro or physio when you take those courses. Today, we're talking about codominance. And so the genotype for type O, it's little I, little I. The I has to do with the enzyme involved. Don't worry too much about that. But it's little I, little I. For type A blood, it's this capital I with a superscript A and then a little I, or big I with the A and another big I with the A. So it's kind of like homozygous dominant versus heterozygous, but there's not really a recessive allele in this situation. The recessive allele, quote unquote, is lacking the marker entirely. But I want you to focus on this codominance concept, which we'll emphasize at the bottom here with the AB blood. But type B blood is going to be B, or this as well. Yeah, for blood type, they just denote the genotype in a different, with these I's, it's a little weird. But essentially, the only way you have type AB blood is if you have both A and B. that genotype. And you see both alleles equally expressed. Both are dominant. Okay, so there are some practice questions, but I think we're going to leave that to the end if we have time, because I really want to cover sex-linked traits, and we'll do this quickly as well. But I do want to get to X chromosome because the lab does talk about that, and I think it'll really help. So let's talk about incomplete dominance first. The lab also addresses this. So we have codominance, and then we also have something called incomplete dominance. Similar, but definitely different. So incomplete dominance. Okay. Here we go. Incomplete dominance is when heterozygous individuals show a phenotype that is a blend or a blending of both the dominant, I'll underline heterozygous too, the dominant and recessive traits or phenotypes. Okay. So what's going on in this example problem here? The genotype here for this flower is big R, big R. And it's crossed with little r, little r. Big R we'll say is dominant. Red is dominant. And so the F1 generation, I'm just going to do the cross. I said it was, the red was big R, big R. The big R is dominant, and dominant is red. Crossed with little r, little r. Similar to the purple and white flowers we did. But this time in the F1 generation, we get that same result. They're all heterozygous. But if the problem says this trait is incomplete dominance, then that's going to produce a blending, right, of the dominant and recessive traits. So when you blend red and white, you get pink. So 100% pink offspring. So Christine, I think I saw your question. How do you tell? You really just have to, it'll be in the problem. You'll never have to guess because you can't tell unless it's in the problem given to you. It'll tell you if it's incomplete dominance. In real life, gets a little tricky. Some traits are just by classic Mendelian genetics, dominant recessive, and it's just, that's just how it is. That's what we see. And and that dominant allele will be expressed entirely if you have one copy. But then some traits have this blending of sorts where both the dominant and recessive contribute to that phenotype. Okay, so I know I'm kind of going quick towards the end here, but I really want to talk about X-linked traits. And that's our last thing to talk about for tonight. But we're going to talk about X-linked conditions, X-linked genetics, where there are genes located on the X chromosome involved in some important things, such as hemophilia. There's a blood clotting gene. So hemophilia is when your blood doesn't clot properly, you keep bleeding out. The X chromosome, there's also a gene for muscle development. So you can get muscular dystrophy if there's a mutation in this gene. And also color blindness. So it's kind of interesting that a sex chromosome, remember it's, you know, in male, female, XY, or XX, the X chromosome, really in contrast to the Y chromosome, a lot of it has nothing to do with determining sex. It really doesn't. There are other genes totally unrelated. And so it's a very important chromosome. It has important genes on it. Okay, so let's look at the genetics of X-linked recessive traits and conditions. All right, let's write some fun stuff here. So genes found on the X chromosome expressed differently in males and females. Because the sex chromosomes, X and Y, are different. As I said, women, female, 2 X chromosomes, right? Males, 1 X, 1 Y. So let's take a look at how this can manifest depending on gender, on your sex. It's the only time, right, that we've ever talked about genetics that are dependent on that kind of thing. It's kind of interesting. Okay, so let's do male on this side, genotype, phenotype. And our example that we're going to use is color blindness. This is a sex-linked trait or X-linked trait, same thing. And then we're also, in a minute, going to do the genotype and phenotype for female. And you're going to see how it can differ. Okay. So for males, you have 2 options. Remember, this is X-linked, okay? So we're only talking about the X chromosomes, not the Y. So for males, you can have a normal X chromosome and your Y chromosome, or an X chromosome with the color blindness allele on it, this little C, and then your Y chromosome. We'll come back to that. Female, you can have 2 normal X's, 1 colorblind X, and then a normal X, and then 2 colorblind X's. How does this manifest and why is it a big deal? Let's look. Normal X, normal Y in the male. You're gonna have normal vision. Correct, not colorblind. Now this one defective X here, or colorblind allele X, will produce a colorblind male. Now this is fascinating because males, you only need one allele for that trait to have the condition. Now take a look at females. This is normal, right? 2 normal copies. Normal, female. This is also normal female. If a female has a functional X chromosome, a functional gene for color blindness or vision, it actually overrides. So it doesn't matter if they have one mutated version. They need to have both to be colorblind. This is actually why X-linked traits are more prevalent. They're more common among males because we only got one shot. We only got one X chromosome. So if it has that version of the gene, if it has the disease version or the disorder version, then you have the disease. You only need one, right? We only got one. But with females, you can have one, but if you have a normal copy, you'll have the normal phenotype. Okay, glad I got to talk about that. We can do the practice problem next class, but I think this will be sufficient for you for the lab. I think this will help for sure. Okay, so 6:50. So we know everything to do. Yes, you do. We covered everything you would need for the lab.